Hey guys, now that you know something about proteins and protein structure, we're ready to move on to enzymes. Now, basically all the enzymes that we're going to talk about are going to be proteins. And so all that stuff we learned about structure, that's going to apply to these things. It's a chain of amino acids folded up, sometimes multiple chains. They've got primary, secondary, tertiary, and quaternary structure. Enzymes, though, are a special class of proteins because there are a lot of proteins that aren't enzymes, like hemoglobin. It carries oxygen, but it doesn't really catalyze a chemical reaction. You have structural proteins, you have receptor proteins, and things like that. So let's go back and make sure we know a little bit of chemistry here. Looking at these two reactions, can you answer this question? Which of these is endergonic and which is exergonic? All right, and that's about spontaneity with delta G. We'll talk about delta G some more. I'll give you a whole PowerPoint on delta G. But that's our change in free energy. That's our ability to do stuff. So hopefully you can see that this one requires that energy is put into it, and this one, you get energy out of it. And so this is exergonic, and this is endergonic. So a lot of these exergonic reactions, they want to happen. They're waiting to happen, but they don't just happen, right? They don't just spontaneously, your sugar doesn't spontaneously combust on the shelf. Your firewood doesn't spontaneously combust without having it get ignited. The reason those reactions don't proceed on their own is because of activation energy. The activation energy has to be overcome to get the reaction rolling. So we have to provide a match to start a fire, right? And that gives us a little bit of activation energy for a little bit of wood. And as that wood begins to burn, it goes through that combustion reaction. We overcome that activation energy, and it releases energy that can then be used to overcome the activation energy of the next molecule and the next molecule, and that continues. And it's also releasing enough energy, one, to provide activation energy to continue the burning of more molecules, and two, to emit light and heat, which is what we call fire. But of course, we can't provide that level of activation energy to break down our carbohydrates, right? That's not going to be healthy. We can't just eat a meal and chuck a match down there and have it, blow up inside of you. That's not going to work. That's too much energy. That's going to be damaging to tissues and cells. So we need a way to lower that activation energy. And that's the job of a catalyst. Catalysts reduce the amount of activation energy. So we go from needing this much energy to only needing this much energy. So instead of all this, we only need to provide this. And this new activation energy, with the presence of a catalyst, this activation energy right here doesn't require a match. It just requires body heat. Body heat, it provides that activation energy, no problem. So if we can lower the activation energy to where body heat is sufficient to have the reaction happen, the reaction will happen. So we can basically make a reaction happen by presenting the reactants with a catalyst. And this is what enzymes are. So enzymes are biological catalysts. And like I said, they're almost always proteins. There are some RNAs that will act as catalysts. There are also protein RNA complexes that act as catalysts. If you think about it, that's what a ribosome is. And there are other things that do similar things. But enzymes are biological catalysts. They lower activation energy. And simply by activating enzymes or providing enzymes, we can make the chemistry happen that we want to happen. We can choose what reactions occur by providing the appropriate enzymes. And since enzymes are proteins, they're coded for by DNA, so we can express genes to make certain chemistry happen. And this is how, your neurons are different from your pancreatic cells. It's because we have different proteins active in there. We've activated different genes. So the proteins are making some reactions happen and not others. And it's different for each cell type. So each cell can be very specialized. Not only that, but we can have it happen in different components of the cell. So let's get some vocabulary on these enzymes first. Like I said, it's generally a protein that acts as a catalyst. That's an enzyme. The thing that the enzyme works on is the substrate. It's the reactants of the reaction that is catalyzed by the enzyme. That's what substrate is. So the active site is on the enzyme. It's the part of the enzyme that binds to the substrate. And so if you've got an enzyme like this one, this looks like this is probably sucrase. It's an enzyme that breaks sucrose. This looks like sucrose, just the way they've drawn this. These two sugars, it's a disaccharide. It looks like fructose and glucose, which is what sucrose is. And so that makes this purple enzyme here, sucrase, the enzyme that breaks sucrose. It has an active site, which is where the enzyme binds that sucrose. It's the part of the enzyme that binds the sucrose. And what happens in this case, it's going to grab a hold of that sucrose, and by binding that, it's going to make it change shape. Anytime something binds to a protein, the protein changes shape. If you stick, even if it's just an electron, a proton, any molecule, you stick something on a protein and it's going to change shape. So you stick the substrate on the protein and the protein changes shape. And it changes shape in such a way that it stresses this bond right here and makes it so that bond very easily breaks. And so when you break that bond, you now have two products. And the products are not the molecule that binds to the active site. So the protein's immediately like, the enzyme is immediately like, get out of here. You're not my substrate. And they get rid of it. And then they're back to their original shape because they let go of that substrate. And now they're ready to receive another substrate so they can do it again. And this is a key characteristic of catalysts, is that they don't get used up in the reaction. They are immediately ready to do it again. So enzymes are reaction specific, which you could probably guess what that means. It means that they do one thing and one thing only. How are they affected by the reaction? They're not, right? At the end of the reaction, they are going to be there again, ready to do it again. They may be changed in the middle of the reaction, but by the end, they are back to their original form. How are enzymes affected by cellular conditions? Well, these are proteins. So what kinds of things affect proteins? The pH can affect the shape of a protein. The temperature can affect the shape of a protein. We've talked about that, right? The amount of salt, anything that can cause a protein to denature or to change its intermolecular attractions from one strand to another, that change that 3D shape. You could stick a phosphate group on there, or you could take a phosphate group off of there. You can attach things to it or take things off it. Anything that's going to change its shape because of the cellular conditions can change its function. And usually this is how we turn them on and off. When we name enzymes, they usually end in ASE. So sucrase, we were just talking about, it's the enzyme that breaks down sucrose. DNA polymerase is an enzyme that makes a polymer of DNA, so it polymerizes DNA. It builds that double helix. Proteases break down proteins, and there's a wide variety of those that break down in different ways. Lipases break down lipids. Helicase is an enzyme that unzips that double helix, right? Separates those two strands. Ligase is an enzyme that ties things together. The word lig just means to tie or to bind. So ligase ties two nucleotides together when you're assembling DNA. We'll talk about every one of these enzymes at some point. So this is just a different diagram showing the same thing. Here's our enzyme with the active site. And again, it looks like it's the same thing. Yeah, this is sucrose again. Different diagram, same thing, similar colors. Sometimes this is described as a lock and key model. It's kind of an older model. It's simpler. Maybe it's more of a freshman year type of model, because what we talk about with the lock and key is that it's about the physical shape, right? The reason the key works in a particular lock is because the key and the lock have corresponding shapes that allow them to be compatible. And they're very specific. You can't use a different key in a different lock, right? You have to use the exact correct shape for a lock to be unlocked by a particular key. So shape is a factor too. So all of this kind of makes it, it's like a lock and a key. It's very specific. It's about its physical shape. Sure. This drawing, I'm sorry for this. This to me always looks like a dragon's head, and kids kept saying, I don't see a dragon's head. What are you talking about, Mr. Powell? So I had to draw the dragon one time, so I drew the dragon. And then later I drew a dragon with a mustache. I'm not really sure why I did that. I think it was like a Mr. Clumpy reference or something. A more sophisticated model than the lock and key model is the induced fit model. It really is about the changes in the shape. We need to be thinking about how when the enzyme binds the substrate, the enzyme changes because of that. And that change is what makes it act on the substrate. It will make it stress bonds, like I was talking about before, or it will put two things together in such a way that they're very likely to react. I mean, think about collision theory. Two particles, to react together, they have to run into each other with the correct orientation with enough energy. An enzyme can make that happen. They can grab one, grab the other, and just immediately they change shape because they have bound these two things in such a way that it shoves them together and forms that bond. So this is what we mean by induced fit. It emphasizes this idea that the enzyme changes shape as it responds to the presence of that substrate and makes that reaction happen with that shape change. So induced just means caused, right? And so when we say it's an induced fit, like it's available, ready to receive the substrate, and once it gets it, it moves into a shape. That substrate causes it, it induces it to go into that shape that makes the reaction occur. So I've kind of answered both of these. Generally, we're either putting things together or we're taking things apart. We're sticking something on something or we're taking something off of something. I mean, we're breaking or we're building. That's the two kind of ways we make things and use things in chemistry, in biology. So with synthesis, like I said, they position the substrate in such a way that they are able to bond together. With the digestion, they position the substrate in such a way that it stresses the bond that's going to break and makes it so that it separates. So I want you to pause the video here and think about all the things that would affect enzyme function. I kind of buzzed through a few of them just a minute ago. You should be able to think of as many things as possible, whether it's, how much, whatever, anything you can think of. I want you to see if you can come up with a list of things that affect how quickly and how well an enzyme does its job. So go do that. Instead of just writing a list, we're going to go through some of these things. The first one is enzyme concentration. Now, we got to be careful with these graphs because it's easy to think this is time and this is a reaction progressing. That is not what this graph is showing at all. This is enzyme concentration. So if I have just a little bit of enzyme, we tried that test, we get this data point. I try a little bit more enzyme, I get this data point. I try more enzyme, I get this data point, right? And if I try lots of different amounts, this is the curve that I'll generate. So it's not time down here, so be careful with that. You want to make sure, in AP especially, you always want to be reading what the axes are. Don't assume that the graph is what you think it is. Read the axes every time. So if you look at this one, at low enzyme concentration, the reaction rate, this is how fast the reaction goes, is pretty slow. And as you add more and more enzyme, try it with more and more enzyme, it gets faster and faster. It goes higher and higher up. And that's all as you would expect. But then it starts to flatten out. And the reason it flattens out is because even if you add more enzyme, we're not adding more substrate. At some point, every substrate has been grabbed by an enzyme, and having more enzymes doesn't really help because there's no substrate for them to work on. So it plateaus, it flattens out up there where we have basically used up all the substrate. All of the substrate is grabbed right at the beginning. We are going super fast and there's no more substrate left. You get a similar graph when you look at substrate concentration, only this time, instead of there being all the substrate grabbed up, the enzymes are overwhelmed. You've got a specific amount of enzyme, right? And as you add substrate, that amount of enzyme is going to encounter the substrate more often. And so it's going to go faster and faster. But at some point, the enzyme is going full speed. It is catalyzing as fast as it can. And the substrate just keeps coming. And it's so concentrated that it doesn't have to wait to find the substrate. The substrate is right there. That's as fast as the enzyme can go. So we plateau again. And that should make sense. I don't think that's too complicated. Things get a lot more interesting when you get to temperature. What I want you to do here, I'm going to have you pause the video again. I'm going to play that little clip again. I want you to explain why this tails off in lower temperatures and why it falls off at higher temperatures. So pause the video, do that, check with the neighbor too. So hopefully you were able to think about this lower temperature thing, right? All chemistry happens more slowly at lower temperatures. There's a couple of reasons for that, right? I mean, we've got, when you study collision theory in chemistry, which is actually what we're doing in chemistry right now, at lower temperatures, things collide less often because they're not flying around so fast. So there's fewer collisions, so fewer reactions, and they collide with less energy, meaning it's less likely for them to overcome that activation energy, less likely for the reaction to happen, so it slows it down. So just by being colder, all chemistry slows down at lower temperatures. Now, the high temperature, the way this drops off like a rock, right? That just falls off a cliff. That's denaturation. That's the protein losing its shape. Remember, when we're looking at enzyme activity, we're talking about proteins, right? Enzymes are proteins. So proteins lose their shape at high temperature. They fall apart. They unravel. They don't tail off and get a little bit worse and a little bit worse and a little bit. They just stop working entirely. They do drop pretty quickly, but then they just completely stop. 37 degrees, hopefully you know the significance of that, but that's basically 98.6. That's body temperature. And there's a lot of enzymes that work really well right here. Of course, lots of them in our bodies. And there's some interesting ones. There's some interesting exceptions to that. And some are maybe a little weirder than others, but this is what a scrotum is for. I know that's kind of awkward and weird. And I don't think I've ever recorded this in video before, so don't spread it around. But basically, some of the enzymes that are used for making sperm in the testicles, they have a lower temperature. They actually work better at like 36 degrees or 35 degrees at a lower temperature. So their peak would be, shifted a little to the left here. And so by hanging out there when it's hot, they hang out there farther. It actually is able to adjust. I should not use my hands for this conversation. But it's able to adjust. It'll shrink or expand based on temperature. And when it's hot, it'll expand and allow for it to get away from your body heat a little bit to kind of cool it. And then when it's cold, it'll huddle up. It'll get smaller to keep it closer to the body, to keep it right in that peak temperature range. Now, why don't we just evolve enzymes that work better at 37? Well, we don't get to choose, right? This was the solution that came up first, and it worked, and it got passed on. Right? It made it more likely to have viable sperms. This is not an easy conversation to have. Let's think of another example. Bacteria that live in hot springs, right? Their activity peak is much higher, and they don't denature so well. So they often taper off at high temperatures and don't actually drop off in denature until very high temperatures because, they can handle water that is nearly boiling. If you look at a typical bacteria that infects humans, their enzymes make a graph that looks a lot like this. And so this is why you have a fever sometimes, or why, even with just a small injury, that area gets hot. It gets hot because mast cells release histamine. You get a fever because it's more of a brainstem thing. It tells your whole body, let's crank this up a few degrees. That'll slow down bacteria. It actually makes the enzymes and white blood cells work a little better. So they have a curve that's a little shifted this way. And so they work a little better at high temperature. So they're not running around being overactive and attacking your stuff normally. They get really active where it's hot. And so you make it hot where you're infected, where you have an infection. And then they get more active. So it's really kind of a cool trick to kind of activate those white blood cells in either a local place or, like I said, in the case of a fever, all over your whole body. If you get a major infection and it's all over, your body will just go, out of hell with this. The small responses are not working. Let's turn the whole thing up. So it's interesting. These peaks of temperature get a little bit more, sophisticated. And it's interesting to see the different enzymes that have evolved to be more effective under certain temperatures. Right. So here's what I was just talking about. Human enzymes around 37. 70 degrees Celsius is not an uncommon temperature for hot spring bacteria enzymes to work great at because that's how hot it is almost all the time there. And they can still work even when it's almost boiling. This is really hot. We will actually exploit an enzyme like this second semester when we do PCR, where we'll amplify your own DNA. We'll take your DNA and make multiple copies of it using an enzyme from a bacteria from a hot spring. It's a pretty cool thing. We'll do that second semester, hopefully. pH is another one of these examples. Different enzymes are going to be more effective at different pHs. Your blood is a little bit over 7, about 7 pH. So, this red peak here is probably any enzyme that works in a typical cell or in your blood. See if you can guess where this and this would be. So I'm going to have you pause the video again. I'll play that clip again and tell me where those enzymes are probably from. So this low one should be fairly easy. Where's the place in your body with a big acid bath? That's your stomach, right? So this is pepsin. Pepsin is an enzyme that works in your stomach. It is activated by the hydrochloric acid that's in your stomach. You actually make it in an inactive form. What pepsin does is it chops up proteins. So you don't want that active inside the cells because it'll start chopping up your proteins, and that would be bad. So we make it in an inactive form. We make it at pH 7, and it doesn't do anything. It doesn't digest your proteins. You dump it into the stomach where the pH is, 2.5, and it swings way down here, and now it's active, and it'll chop up proteins. So it's only chopping up proteins when it's in your stomach. It can't chop them up when it's in the cells where they're made. It's kind of a cool trick. This other one here, this blue one, this is trypsin. Your pancreas here produces bicarbonate to neutralize the acid coming from the stomach, and it dumps right in here at the beginning. So once you get to here where you're starting to really kind of digest things, the pH has been neutralized. In fact, it's a little over neutralized. It swings a little bit high, a little past 7, up into the 8-9 range. And that's where trypsin is working best. And there are other digestive enzymes that work really well there that have a peak a lot like this. Salinity is a similar type of thing. With a lot of salt, it interferes with the structure of a protein. I think I mentioned that when we were talking about denaturing proteins. It interferes with hydrogen bonding. Anything that requires a charge is going to be messed with. So it's going to affect the shape of the protein. Low salinity is actually pretty bad too because of the same thing. It's going to make different interactions occur. We have a lot of compounds that help with enzymes. We divide them into a couple of different categories, and you might hear these terms used a little bit differently and sometimes even interchangeably. I've found all kinds of, it's honestly kind of confusing terminology here, but cofactors, typically we're talking inorganic compounds. It's magnesium in chlorophyll. It's these different ions usually they're metals that are key to making the final shape of that protein. So in addition to being, tertiary, quaternary, sometimes they need an additional thing there. Like I said, hemoglobin won't work. If you don't have the iron, you're not going to be able to make the final product of hemoglobin. There's no amino acid with iron in it. Iron is added later. That's a cofactor. If those cofactors are organic molecules, we call those coenzymes. And these are usually what we call vitamins. So these two together, these are vitamins and minerals, right? These are things you have to eat. They have to be in your diet if you can't synthesize them. A lot of the coenzymes you can synthesize. Ancestrally, we used to be able to make vitamin C, for example. Our ancestors could make them. Most mammals can make vitamin C, but we lost that gene in the same way that those cave fish lost their eyes because we ate so much fruit. Not being able to make vitamin C was not a problem. It was always in our diet. We didn't really notice that it wasn't in our diet anymore until we started taking long trips in the ocean where we couldn't encounter fresh fruit. And then we got scurvy. These are like the last little tweak you have to put on the enzyme or the protein to make it function properly. So primary, secondary, tertiary, quaternary, and then sometimes cofactors. And sometimes we call these activators. They're things that make the protein work. And sometimes we use them as a switch to turn on the protein. What we usually use to turn on and off proteins is inhibitors. We have a couple of different types of inhibitors. We have competitive inhibition and we have non-competitive inhibition. A competitive inhibitor gets in the way of the active site. So this purple guy is the enzyme. This little light blue guy here, that's the inhibitor. And it gets into the active site so that the substrate can't bind. And it'll get in there and it'll get back out and it'll get in there and get it back out. And so it'll slow down the access to the substrate. So it'll slow down the reaction. So inhibitors can do that. We do have some toxins, some poisons that we'll talk about in a minute that get in there and stay there and won't move out of the way. But that's irreversible inhibition. It's not really something, it's something we want to avoid. But you can overcome this by increasing the substrate. If you've got just a little bit of these inhibitors and a whole bunch of substrate, then when this enzyme goes to grab something, it's almost always going to grab the substrate. And therefore, it's going to catalyze the reaction. And the reaction rate will be nice and fast. It's only a problem when the amount of inhibitor is high compared to the amount of substrate because it's grabbing the inhibitor and wasting time on that. And we'll see that in some data that we're going to look at later. Sometimes we use these as medicines. Penicillin blocks an enzyme that bacteria use to make cell walls. And if they can't make cell walls, then they can't reproduce. There are other examples. Non-competitive inhibition, we usually call those allosteric inhibitors. So allosteric just means other location. So it's binding not in the active site, but when it binds somewhere else, it changes the shape of the enzyme so the active site doesn't fit the substrate anymore. So just by binding somewhere else, it pushes the active site into a different position so that now we can't bind the substrate. And so this is usually a way that we can just shut off an enzyme. So we can make a whole bunch of enzyme, have it ready in a cell to do something, but not do that thing until we turn it on. So we can have some other enzyme come along and its job is to remove these inhibitors and it pulls the inhibitors off and now all these guys are active. So it's a way to kind of control exactly what's going on inside the cell. We do that by first having specific enzymes and two, activating and deactivating specific enzymes. So we can very carefully control exactly what chemistry is happening inside of a cell. We use these in technology as well, cancer drugs, things like that. And there are toxins that work this way too. If it's a toxin, usually it's an irreversible inhibition. It shuts off the protein completely and makes it so the protein can't do its normal job. When we were looking back here at this allosteric cyanide, right? Cyanide interferes with cytochrome C, and that's in the mitochondria. It's part of the electron transport chain. It's part of what allows us to get ATP from food. And so if cytochrome C gets knocked out by an inhibitor, by cyanide, then you're not going to be able to produce ATP. And if you can't produce ATP, you get dead real fast. Okay, so irreversible means it binds and doesn't come off of there. You can be permanently binded to the active site. You can be permanently bound to the allosteric site, some other location, and that's fine. Either one. They can be both competitive or allosteric. It doesn't really matter at this point. You've lost the ability of the enzyme. So when we activate or deactivate proteins, we are changing the protein's shape. Remember, when we change shape, we change function. And we can do that by sticking absolutely anything on there or by taking absolutely anything off of there. Here's what I mean. You can stick an electron onto a protein and change its shape. You can make it work. You can make it stop working. You can add some other atom. You could stick a calcium on there and it will work or stop working depending on the enzyme, right? We have different ones that work in different ways. A lot of times what we use is phosphate. You stick a phosphate on there. You get a phosphate off of an ATP, right? You take an ATP, you take that phosphate off, you get energy. You also get a phosphate that you can stick onto something and make it active or make it inactive. You can sometimes pull off a molecule and make it active or inactive, right? So anytime you stick anything onto a protein, you can make it active. Anytime you stick something onto a protein, you could make it inactive. It depends on the protein, right? You can do the same thing by taking it off. You can either take it off and make it inactive or take it off and make it active. So by sticking or removing things on proteins, you change what they're doing. A lot of times we put these enzymes physically in a membrane to make an assembly line. You think it was Henry Ford? It wasn't Henry Ford. Your cells have been doing this for billions of years. So we can take this guy, which is the substrate to enzyme 1. Enzyme 1 makes this, and we stick another enzyme right next to it that does this, that turns it into this, and here's the thing we need. So we make these metabolic pathways, and we assemble them right into our cells side by side, so you just pass the substrate right down the line and make our final product, even though we need a whole bunch of enzymes to make that happen. Very efficient, very well controlled, allows us to do, complicated things quickly. This is the electron transport chain I was just talking about. There is cytochrome C. We were just talking about if you interfere with that, right, you can't pass electrons, which means you can't pump protons, which means you can't generate ATP. We organize these things together in such a way that we can couple exergonic with endergonic. We can make it so that we get the energy from one reaction and use it to do another reaction. We do this kind of thing all the time. We'll talk a lot more about this when we do cellular respiration and photosynthesis pretty soon. They both have an electron transport chain that's like this. And that's coming up. We also need to talk about feedback. This is something you probably have some idea about. We've kind of talked about when we talked about homeostasis. We use negative feedback loops. Here's a metabolic pathway where I'm making product G. And product G, when I get a lot of that, that's the molecule I need for whatever purpose. Product G is actually an inhibitor for enzyme number 1. So I stop turning A into B. So I stop wasting all my energy making more G when I have plenty of G. Now, when I'm running low on G, I'll be like, I need some G, and I'll take the G off of this enzyme 1, and suddenly enzyme 1 is active again, and now it's going through this whole process again. So we can do feedback inhibition. We can shut off the whole assembly line with the product, using the product to do that. Here's a perfect example. Here's isoleucine. This is an amino acid. You need this to make proteins. When you have lots of isoleucine in cells, you don't need to keep making it. And so isoleucine will bind to an enzyme that is early in this process and stop making more isoleucine. And again, when you need isoleucine, you'll be looking around for one. You'll grab that one off of there. Suddenly, this guy is active again, and we start making isoleucine again. Pretty cool, simple little solution. Prevents us from overproducing any particular molecule. Got a little macromolecule review for you. I want you to pause the video and I want you to answer every one of these questions. So here we go. All 4 groups of macromolecules have carbon and hydrogen. I can't really think. I guess maybe they all have at least a little bit of oxygen, but all have lots of carbon, lots of hydrogen. What types of macromolecules always have nitrogen? You always have nitrogen in nucleic acids, which we didn't really talk about. We'll save that for later. You always have nitrogen in proteins in that amine group, right? Sometimes you find nitrogen in lipids or carbs. Chitin, for example, is a carbohydrate that we talked about in the exoskeleton of arthropods, or also in the cell walls of funguses, and that has nitrogen in it. There's probably steroid hormones with an amine functional group on there. Which type of macromolecules usually have sulfur? That's proteins. They form those disulfide bridges, cysteine, and there's another one that have sulfur in it. Which types of macromolecules usually have phosphorus? That's your nucleic acids. They have a sugar, phosphate, and nitrogenous base. That makes up a nucleotide, which is a monomer of DNA or RNA. You don't need to worry about that one at this point. Sometimes you find phosphorus in other ones, like a phospholipid. When we're looking at cell membranes, we've got the two fatty acid tails attached to the glycerol, and then there's going to be a phosphate and some other things stuck on there that give it that hydrophilic head. But that's the only lipid that has that. So hopefully this wasn't too bad. Mastering is going to be available. So if you are looking for something to do for biology, right, you can start working on this mastering. Hang in there, guys. You're doing great.